Hello and welcome back to AP Psychology. I'm Miss Lee and we are in Unit 1 Scientific Foundations. Today we're going to talk about experiments. If you haven't done so already, go ahead and grab your notes. Okay, we have a lot of learning targets in this video. We are going to go over a lot of vocabulary. It's very important vocabulary. Sometimes it can get a little tricky and people can get things a little bit mixed up. But I am going to try to keep this under half an hour, I promise. What we're going to be talking about is how we set up an experiment, the different factors of setting up an experiment, and the reasons why we do what we do in experimental research. Okay. So first we're going to talk about cause and effect. In our last video when we were discussing correlation, you might remember that I said that experimenting was the only way that you could say that one factor caused another. So like in other sciences, experimentation forms the backbone. It really forms the foundation of research in psychology. Basically what that means is you can't observe your way to a hypothesis. You can't survey your way to testing out a theory. You have to be able to say conclusively if one thing causes another. So some examples of famous experiments in psychology: Pavlov's salivating dogs. We'll talk more about this in the learning section, and this picture on your slide depicts that experiment. Milgram's obedience study, which we will be discussing in social psychology unit. Ash's conformity experiment, which will also be in the social psychology unit. Experiments are the only research method that isolates cause and effect. So everything that we've talked about up until now will give us good information, but this is really the only way that we can conclusively say cause and effect. If you have not written that down on your notes right now, go ahead and write that down. Underline it, highlight it. It is critical that you understand that. On the AP Psychology test, you want to be able to get answers correct if they are asking if a correlation shows cause. It does not. You can go ahead and say no. That is a very important thing when you're talking about experimental research. So we're going to kind of set up a hypothetical experiment and walk our way through each of these terms on your note page. It's a big chart. You see definitions of these terms are written down for you. They're already there. But what I want you to do is come up with an example of your own. You can also jot down the example that we're talking about in this video, but I would really like for you to think about an experiment of your own. So first of all, you have a hypothesis. This is a testable prediction, an if-then statement. If teachers at our high school drink coffee, drink caffeine, they will stay alert and happy. That is a hypothesis. It's a testable prediction. So what do we want to do? We want to test the effects of caffeine on the behavior of high school teachers. We want to look and see what effects caffeine cause in these teachers. At this point, researchers want to create operational definitions for the variables in their experiment. What does giving teachers caffeine actually look like in real life? And how are we going to measure alert and happy, those variables in our hypothesis? From our last set of notes from the last video, what do we use to measure attitudes, beliefs, preferences, feelings? Hopefully you have come up with, we will use a survey, and that's going to measure the teacher's state of happiness, how alert they are, all of those things that we can't see directly, we can't measure directly. Then you want to ask, who is your population? What is the larger group or category of people that you're researching? Well, we're researching teachers, more specifically teachers at our school, in our regional area. So our population is teachers at our school, but we can't really realistically do an experiment, ask a survey of all of the teachers in our region, all of the teachers even in our school. There are a lot of teachers at our school and in our region. So what do you want to do? You want to take a sample. A sample is exactly what you know of as samples. If you ever go to the grocery store or you go shopping with your folks on the weekends, sometimes if you're really super lucky and you go to the cheese section, you get those little tiny pieces of cheese. They aren't going to give you a great big block of cheese. They're not going to give you the population of cheese. They're going to give you a tiny little slice, a tiny little fraction, a sample of the cheese. Now, hopefully the idea is that it's going to represent the whole block of cheese, and you can decide whether you like it or not. It does need to be a representative sample. Hopefully you don't get the one tiny little piece of cheese that has the big giant jalapeno in it. Or if you're sampling chocolate chip cookies, it's the only piece of the chocolate chip cookie that doesn't actually have a chocolate chip. That's not going to be very representative of what a typical cookie or a typical piece of cheese from that block is going to be like, what the experience is going to be like. So hopefully your sample is going to be representative of all of the people in the population. What would that look like in our area? So you want to make sure that we obtain a representative sample. You want to make sure that you are getting a lot of different types of people, a lot of different types of teachers, not just teachers from the math department, for example, not just teachers from the art department, for example, not just male or female. You want to make sure that your sample is representative. There are types of samples. The first one is a random sample. In a random sample, everyone in the group, the larger population, has an equal chance of being chosen for the experiment. So for our experiment, how would we do that? We would pull names from a hat. We could probably just put names of each of the teachers in a hat and draw out the number of names that we want to be in our experiment. And it depends on how big your population is. If you want half of a sample instead of a full population, if you've got 100 teachers, you could sample 50 and just pull 50 names out of the hat. That should be fairly representative. Another way of sampling is to get a stratified sample. What you want to do in a stratified sample is you want to divide your population into subcategories that make sense to you. So you want to make sure that each department is represented in a way that is representative of how many teachers is from that department. So if you have, out of 100 teachers, you have 10 of them from social studies, you want to make sure that at least one person from social studies is represented in your sample. Once you have divided your population into the relevant subcategories, then you pull a random sample. So then you make sure that you pull one name from the subsample of subcategory of social studies. So how would we get a stratified sample in our population? Well, we want to divide our staff into categories: male, female, what subject they teach, whether they're new or veteran teachers. Those are examples of how you could get a stratified sample. Key understanding: we randomly sample so that the sample is representative of the population. Why do we do that? So that we can generalize our findings, so that we can say, yes, this represents our population. We can say something about the population of all of the teachers at our school. We can say something about them because we have pulled a random sample of teachers for our experiment. Very important that you understand why we randomly sample. Next, you randomly assign. Once a sample is obtained, the researcher randomly assigns participants to the experimental or control groups. Okay, so you've got your participants in your experiment. What it means to randomly assign is you basically put people in either group, the experimental group and the control group. Assigning participants to an experimental and control condition by random assignment minimizes pre-existing differences between the two groups. How do we do this in our experiment? Well, after the participants have gathered, we could hand out numbered cards in no particular order to separate all into control and experimental groups. So our key understanding here is that we randomly assign in an experiment so that any pre-existing differences between groups does not impact the results of the experiment. It should kind of wash out, so to speak. It should kind of even out. What could pre-existing differences be in our experiment? It could be caffeine tolerance, or experience with caffeine at all, activity levels, overall health, anything that makes those differences between people. Some people don't drink caffeine at all. Some people drink lots and lots, so they have a higher tolerance. So you want to make sure that you are randomly assigning. You don't want to take volunteers, because what if everyone who volunteered for caffeine has a really high tolerance for caffeine? So you want to randomly assign. All right, so let's go back a step and talk about the different groups. Our experimental group, this is the group that receives the special treatment. I call it the thing. So your experimental group is going to get the thing that you are looking at, to see if it makes a difference, to see if that thing has an effect on a behavior or a mental process. In group A, this is our experimental group. Participants drink 2 cups of regular caffeinated coffee every morning for a month. That is our experimental group. We have randomly assigned half of our sample into the experimental group. They're going to receive the special treatment, the thing. Our control group, or groups, are the folks that don't get the special treatment. They don't get the thing. And we can have more than one control group. So let's say group B is our participants that drink 2 cups of decaf coffee every morning for a month. Group C participants drink hot brown coffee flavored water for a month. So it's not coffee, but it's just water that's been flavored like coffee. Our variables. Okay, here comes some really important vocabulary words. The independent variable. Don't get confused. The independent variable is going to do its thing no matter what. It's the thing that causes, in theory, whatever effect or mental process you are hypothesizing about. It's the factor that's being manipulated by the experimenter whose effect is being studied. You want to see what that thing does to the teachers. It's the type of drink. So caffeinated, decaffeinated, or that brown coffee flavored water. It's really important that you understand that everybody gets the independent variable. It's just a difference in degree of how much or what type of variable they're getting. And the dependent variable is the factor that could change in response to the independent variable. In psychology, it's usually a behavior or a mental process. So you're looking at behavior, either alertness, crankiness. How did we operationally define this? If you think back to, we were talking about happiness. So however we decided that we were going to measure the dependent variable, that's what we're actually looking at as an effect of the independent variable, the thing. Our key understanding here is both groups receive the independent variable. The difference is that they receive degrees. Okay, another huge term in psychology. You must understand what a placebo is. It is the pseudo treatment. It's a fake treatment. Sometimes in studies that we do on medicine, it's basically a sugar pill. It is a fake out. Why do we use a placebo? Well, it's not just to, fake somebody out or play a trick on them. What you are wanting to do is to test out the true effects of independent variable. You can't just say, all right, this group is going to get the coffee. This group just drink water. You're on your own. We're not going to give you anything. Because sometimes the belief of a person in the thing that they're doing can actually alter their behavior. What is the placebo in our experiment? It is the decaffeinated coffee and the coffee flavored water. They are the stand-ins, the pseudo treatment, the fake treatment. They are the things that are standing in for the caffeine, so that the folks who are being tested, who are in the experimental group or the control group, don't know what they are getting. Why is this important? It's this phenomenon known as the placebo effect. This is a film that we're going to watch together, and it will show just how interesting a placebo effect is. There are some very strange things that can happen, and people can actually become addicted to placebos, even though there's not any kind of biological change in the person. Their belief in that thing is so strong that it changes their behavior. Okay, so what are we really doing when we are setting up our experiments? Well, experiments are run by people. So it's important that we control for bias. Sometimes people have a bias. So there are a couple of ways that we do that. The first one is called a single blind research methodology. And in this type of study, only the participants are unaware of which group they're in. So in our experiment, the teachers don't know if they got the caffeinated coffee, the decaf coffee, or the coffee flavored water. They don't know what degree of the variable they received. In a double-blind research study, both the participants and the researchers who are distributing the experimental thing, the independent variable, they don't know either. It is documented somewhere, but they are not aware of who gets what when the person receiving the thing or the placebo is right in front of them. Teachers and the researchers collecting the data don't know who is in what group. Which is better? Well, hopefully you think the double-blind research method is a better way of doing that. Why? Well, because the researchers can be biased as well, whether it's intentionally or not. Let's say that Mrs. Smith is in our research study, and we know that Miss Smith is in the control group. She is not receiving any caffeine. Well, so when Miss Smith is coming to do her survey, as a researcher, if I know that Miss Smith, who usually drinks coffee, is not getting any caffeine, I might give her survey like, oh, here we go, Miss Smith. How are you feeling? Do you need to sit down? Are you tired? It's okay, take all the time that you want. And it's not because I want to sway her, even though I am priming her. That's a term that we'll get into future videos, but it's kind of our natural human nature to take care of our experimental participants. Confounding variable is something that you want to be very, very careful of. These are other items that could affect the outcome of the experiment. They can have an effect other than the independent variable alone, and that is bad. So what could be some confounding variables in our experiment? Well, there is still a tiny bit of caffeine in decaf coffee, so that could throw our results off. Some people have more or less experience with caffeine, and so their experience leading up to the experiment could change the outcome. Even something as simple as the amount of sleep that people get in a typical night, or if they've had a bad morning or a good morning, or something traumatic has happened to them. All of those can be confounding variables that could affect our study. And that is it for experiments. We are very soon wrapping up Unit 1. We have one video left. I can't wait to see you in our next one. Bye for now.